Hi everyone. Okay, so today what we'll be doing is a practical session. Okay, hold on, just let me get my stuff. Okay, when we are back. Okay, so we'll go through our practical session today. This is a three-part practical session. We are actually looking at how to actually analyze microarray data using R. And let me just get the documents up. Okay, so this is what we'll be doing. So these are three things that we'll be doing. First is we have a microarray data set. It's actually from a 2008 publication by Kuhl et al. So in this data set, let's look at it for a little while. This is the publication. So what they have identified is they have 62 samples, patients of medulloblastomas. And what they realized through the analysis of microarray is that they identified 5 different subtypes. So it can be 5 stages of tumor, 5 different stages of this brain tumor. Okay. So what we are going to analyze is, are there actually 5 types of tumor? Okay. So this is the first part. After that, what we are going to do is, because the exact number of the subgroups is not exactly known, so we are going to analyze that. And then the first session, which is now, we will actually identify how many tumor subgroups there are. Subsequently, we are going to identify what are the differentially expressed genes for each tumor subgroup. So for example, from healthy tissue to let's say stage 1 cancer, what are the genes that's expressed from stage 1 to stage 2, from stage 2 to stage 3, and from stage 3 to stage 4. So what are genes that's expressed? So we can actually try to deduce some biological functions. What happened in the biology that causes for cancer to go from stage, let's say stage 1 to stage 2. Okay. Once we do that, then the third session, we are actually going to first generate or create a machine learning classifier to help us classify tumor subgroups. Okay. Having done that, we have a new set of data that we want to test with our classifier. So basically, now is using this Kuhl et al. Data, we are able to classify into 4 different or 5 different subgroups. We will look at it later. Can I actually bring in new data, and then let's say new patient data? You tell me what stage of tumor this patient has. So that's what we are going for the third session. Okay, so in short, what we need is your R. So in order to install R, you have to go and install. Okay, let me just close this. For what I'm using R version 4.0.2. So this is the updated version. And then I'll also install RStudio. Okay, RStudio will make my life a bit easier. So you can install RStudio here. Okay, so I leave this up for you to install. Okay, you can choose your version. Usually I just go for the free version. Okay, so let me just fire up my RStudio. Okay, so my RStudio. Okay, it will launch into this. Okay, so I want to check whether am I using the correct R version. I'm using R 4.0.2, so this is the correct R version. Okay, then what I do is, before I start off, my data is actually in this folder called practical, and in there I have practical 1, practical 2, practical 2, and practical 3. So what I need to go is practical 1, and that is where I get my documents from. Okay. So in R, how do I get that? I have to go to sessions, set working directory, choose directory. Okay. Then I go to my desktop or wherever I have the practical and practical 1. Okay. Then there's nothing else inside because I'm choosing a folder, a working directory. So I click OK. And here you will see that it has been changed. Okay, so this is my files. Okay, so the very first thing that I need to do is to install the libraries that I need. First, I install all the CCRAN libraries. Okay, so let me install it. If you have not installed, this will take some time. Okay, but I've done that already, so it will just be rather quick, hopefully. So you just have to wait for the cursor. So this is the cursor that we're waiting for. Wait for this cursor to be completed. So undergo some installation plots. Hopefully everything is installed well. Okay, so there's nothing much you can do, just wait. Now let's keep installing. So you can put in multiple commands. Okay, so once you see this cursor, this caret sign, it means that it's done. So now you can type in the next command. Okay. Now the next step is actually to install Bioconductor library, but Bioconductor installation has been changed. So if you use this, you're going to get an error. Let me just try and show you how does it look like. Okay, so once I have that, I just press enter. Okay, so now it tells me that error. To use for our version 3.5 or greater, since I'm using 4.0.2 to install, I have to it tells me what to do. So let me go to bioconductor/install, and then this is the page that I have. It tells me how do I install Bioconductor now. So first step, I will need to install the core library. So I've got to run this. So run this. Okay. So run these few commands. Okay, just copy and paste. It will work just as well. Okay. Now Bioconductor is getting installed, so it takes some time as well. Update everything. So you press A and press enter. Okay, so now Bioconductor is installed. So the next step is to install the packages. Okay, so let's go back and look at the documentation. It says that I can install specific package with this command. So I want to install specific package because this only install the core packages. So let me just copy the whole thing. And I just kind of, okay, so these lines are what I've typed in just now. Okay, so now I want to install. See, I'm going to install all these specific packages. So why not I just copy this whole thing over and then just stick it up here. Okay. Then I copy this whole command for installation. Let's enter. While we are doing that, I might as well just save this file. And here I will just say that I will cancel it. Yeah. So these are the new ones I know I'm supposed to use. Save it. Okay. So let's go back to, okay, now it's installing a few other stuff. Bioconductor is a huge package for microarray or bioinformatics analysis using R. Okay, so if you have used Python, you will be using things like BioPython. If you use Java, you'll be using things like BioJava. But Bioconductor is for R. Okay, let's just wait. Nothing. You really can't do anything. Depends on how fast your computer is and the installation steps. You really just have to wait here. So now it's trying to install a few other things. Okay, let's just look through the next step. The next step is after I've installed, I use the library command to load various libraries onto the Bioconductor. Okay. So let me just copy this in readiness. And well, here I just have to wait. Okay, update all. This back port seems to be getting a bit old, so I'm not sure what's happening. Okay, so now it's finished. Okay, so what I have to do is I just paste the whole set of library, and I just want to make sure that nothing is giving me error. Okay, so let it run finish. Okay, just one more. GL, okay, that's the last one. So let us go back and we just see if there's anything that is giving us problem. So FE looks like everything is okay. Library, nothing showing me a big word. It says error, going down, nothing shows me error, and so on. Okay, so things looks okay. Now, I do not have to change my working directory because I already changed. I have already changed my this. I had already changed my working directory in the first place. So I already changed my working directory to begin with. Okay. So the next step is then to load up my microarray data. Okay, loading up microarray data. So what is the data coming from? The data is actually this. Okay, filtered.expression data. Okay, so I can load it two ways. I can use a command, or using RStudio, I will just double click it, and then you ask me whether to load it or not. So I say yes. So once it's loaded, you will see that it's actually loaded in the data. Okay, you can actually double click on this, and then you have a, well, you show how the data looks like. Okay, so you show you a section of data. Okay, so each column is one sample. CL is a microarray data, microarray sample, and then each row is actually a probe. So once you're done with it, just close it. Okay. We can actually use this dimension function, DIM dimension, to find out how many rows and how many columns. Okay. So dimension, let's just see. Okay, it tells us that we have 1,500 rows, 62 columns. So basically, it's a data set, or really a truncated data set of 62 samples, clinical samples. Each sample is a microarray, and each microarray consists of 1,500 probes. So you take 62 times 1,500, you get 93,000, 93,000. Okay, so far so good. So what we want to do is to explore using principal components analysis. So principal component analysis, we put the data into, put it inside a variable called filter.expression.pca. So principal components actually tells us a lot about what are the variations inside the data. Okay, so let us just run it and see what happens. Okay, it's very quick. So now it's finished. Okay, using this precom command. The next step is to plot the data. Okay, let's say we plot the variations for the first few principal components analysis and see what happens. Do this plot command. Not sure if you see it. Okay, plot. So now it plots out a small graph. So this is the first principal component, second, and so on. Now, what is principal components is this? What is that one probe or one gene, in this case probe, that accounts for the most amount of variation? Okay, then it goes to, you actually pick up the probe that has the most variation, then you go to the second probe that has most variation, and so on. So it's a decreasing number of variants. Okay. So what does it mean? Let's just look at the summary. Now remember, the summary, here's what it says. Okay, so let's go up to here. The importance of components. So principal components 1 all the way to 62. Okay, what it says is the first principal components account for 19.7 standard deviation. Okay, so 19.7 standard deviation, you square it, that gives you a variance. But what is very important is this proportion of variance. Now, my 62 clinical samples have variations, but my first principal components, PC1, which is maybe a probe, accounts for about 25.96% of all the variations, while the second principal component accounts for only about 15.5%. Collectively, the first and second principal components account for about 41%. Okay, so this gives us some idea how many principal components do we need? So how many genes how many probes do I have to account for if I want to account for just 90% or 80% of the variation. So we just go to the cumulative proportion. If I just want to bother about 80% of the variation, I only need 18 different probes. Okay, because it's a diminishing return. If I want 90%, I will need here 32, almost double. Okay, ideal if we put everything in, okay, we are going to have 100% for sure. But who wants to deal with 15,000 probes? You may just want to deal with, okay, do I have, let's say if I only cash, if I have only finances to do 11 probes, to study 11 probes, maybe that is a good panel to a good panel of genes to account about 71% of all the variations. So this is what principal components does. Tells us what is the level of variation we have. Okay. The worst you can see, the worst you will have is the first principal component is actually so weak. Then you take a lot of principal components or many principal components to give you the variations, account for all the variations you want. Okay. So, and one property of principal components is that it should not, any two principal components should not correlate to each other. If component 1 correlates with component 3, then you must well just use one of the components, right? Okay. So here we plot the principal components, the first two principal components. PCH19 is just the what you call as the marker. So we just plot it up and see what happens. So you don't, here is what you see. Do you have any is there any correlation? Doesn't seem to have correlations, so that's good. How about we can plot in multiple we want to plot the first 4. Okay, so the first 4 is basically we plot it into a axis, okay, using pairs. So it gives us first 4. Here is how we read. This is the graph for principal component 1 with 2. This is 1 with 3, 1 with 4, and so on. Now there's a limit to how much you can plot in itself. Okay, so you can even any time you can export the data out as a PDF or as an image. For example, I can plot maybe if I want to repeat the command, I just go up, and let's say I want to plot 1 to 6. Okay. Now it will be smaller and smaller. Try not to overdo it because your computer may just crash on you. All right. So this is just a diagnostic tool to help us see whether are things going right in the correct direction. Okay. We haven't actually done anything yet, but very importantly, next step is, can we actually put all the 62 samples onto a heat map? So this is a heat map. We plot it as a matrix, okay, and we just label it with from dark blue, white to dark red. Okay, so the intensity is from the lowest intensity is dark blue, medium intensity is white, highest intensity is dark red. Okay, so let's run this heat map command and see what happens. Okay, what did I do? Yep. Okay, so you have a heat map now. Okay, so the purpose of this heat map, let's just open it up. We have the samples, 62 samples as columns. So the rows is 1,500 genes. So using this heat map, we just want to have a quick visual how many groups there are. That means how many vertical lines you can draw to separate the groups. Okay, we can see that there may be a one large chunk group here. Okay, then this may be one group because it looks very different. The next group will be this one, will be very different. You have some missing. But let's say in this large chunk itself, we find that, okay, we can see that there's somehow a little separating line between here. This group is kind of similar yet different from this group at the end of it. Okay, so it seems that there may be 3, 4, or 5 groups. We do not know, but it's a diagnostic. Okay, so let's go back. So heat map can only show us a very primary diagnostic tool. Okay, so why not we cluster things onto your NMF? NMF is to help us to pick up the biological metagenes. Okay, so we want to run an estimate, okay, using MNF command, and we want to run, okay, show me how things looks like if it is 2 groups, 3 groups, 4 groups, 5 groups, and 6 groups. Okay, so because just now we assume we look the data, it seems to have 3 to maybe 3 to 5 groups. So we want to go 1 plus minus 1 more, so 2 to 6. Okay. It's doing a analysis 50 times, so n run is 50, and this seed tells you that for every, so it should give you the same result for my computer and your computer. Randomizer is never randomized. The seed actually makes it non-randomizable. Okay, so let's run it. This will take some time, okay, because it's running multiple ones. So let's wait. On okay, our session, I have to say allow access. Okay. Okay. So the data is here. Let's wait for us estimation and for it to pop up here. So this will take really a while. Okay. At this moment, you're welcome to go and grab a cup of coffee, or go to the toilet. Yes, there's actually nothing much I can do. I just have to wait. Okay, anyway. All right. So it's finished. I've waited almost 5-10 minutes. I've had my coffee break as well. Okay, so the next thing is we want to see what is inside here. So you use a plot to plot it and see if you can find anything out from it. Okay. So what does it do is it will plot between 2 to 6 groups, and we want to see whether that's the residual, the meaning is residuals, there is an error after each grouping. Does it drop? So we do not see any significant drops here, so it may not be that useful to actually help us to look at it. Now what we have is the dispersion. So group 4 groups and 5 groups seems to have a large difference. So we want to see whether is 4 group better or 5 groups better. Okay. Remember the paper, which is the paper that we have just now, tells us that 5 groups, there are 5 different subtypes. We may think that maybe 4. So let us just look further. We run the consensus map and see what happened. So let's zoom it up. Okay. So this is how you read a consensus map. Let's finish everything first. Okay, one more. Okay, so this is how we read it. This tells us 2 groups. Okay, things are actually well separated for 2 groups. For 3 groups, there may be some errors, but it looks okay. So you want to see when does the diagram actually starts to disperse. So 4 groups seems to be a better one. 5 group it starts to disintegrate, and 6 groups is even worse. So that tells us that maybe 4 groups is better, although we have some errors here. Okay. So that is we're actually battling between 4 and 5. So now we are seems to collecting more evidence that there are actually 4 groups in the data rather than 5 groups in the data, or 4 subtypes in the data rather than 5 subtypes. Okay, let's continue. So here we're actually trying to argue, have more evidence whether it's a 4 or 5. Currently, I'm selecting 4, but I'm comparing 5. Okay, since that's what Koo et al. Selected. So let us run the 4 subgroups first. Okay, we do a filter MNF, and now we only run rank 4. Okay, so this will be much faster because just now we are running from 2 to 6, so now we are just running 4. Okay, so instead of 50 times, we will need to run 20 times. After that, what we do is we have a coefficient map. So we'll see how the coefficient map looks like. Let's wait for this to finish. Even then, it takes a little while. Okay. Okay. So then we have, let's do the consensus map, the coefficient map. What the coefficient map does is, what happens, does it look okay if you have just 4 groups? Okay, so let's zoom out. Is it done? Okay, let's zoom out. So we see the red one, this tells us that this is in group 1, this is in group 2, this is in group 2, this is in group 3, group 4. Okay. Remember, we have a few of fuzziness, so these are the few ones that is giving us problems. So it seems that 4 groups, there isn't much overlap, significant overlaps. So maybe there are 5 groups because of here, these 2. The 4th group maybe actually be split into 5 groups. So we want to see whether is that the case. Okay, let us slowly look at it. At the moment, it seems that 4 subtypes or 4 groups seems to work reasonably well. Okay. Let's continue. And let's predict the assigning. So we actually write the groups. We actually predict the 4 categories into a grouping. Then we write it out into names. Okay. So we actually write out the table. The table is your group assignment.csv file. Okay. So let us just run these few steps at one go. Okay. At the end of it, what you see is in practical 1, there's this group assignments. Okay, that's what we wrote out. So let us open the Excel file and see what happens. So here, these are the microarray data. So there are 62 of them, 62 microarray data. Column B tells us which group it falls under. Group 1, group 2, group 3, or group 4. So this is more of our diagnostic purposes as well. So there's nothing much we can do here. Just view it and see things are actually looking well. You don't save it. Okay. After that, what we want to do is to do a silhouette plot. Okay. So we want to do a silhouette plot based on, let's say, the 2 groups. Okay. So let us, because our numeric groups, this is what we have assigned the groups, and we want to do for 2 groups. Okay. Let's just do the silhouette plot and see what happens. Okay. After that, we have to plot the data. Okay. So once you plot the data, now this is your silhouette plot. So it shows you that there are 4 clusters, 1, 2, 3, 4. Okay. So let us zoom it out, you'll see a bit better. So there are 4 clusters here, but okay, so there are cluster 1, cluster 2, cluster 3, cluster 4. And in cluster 1, there are 26. Cluster 2, there's 9, cluster 3, there's 12, cluster 4, there's 15. This is the one that gives us more problems. Okay, remember, just try to commit to memory first that for 4 groups, there's actually 2 that goes into a negative silhouette. Okay. We want to see if 5 groups, how many negative silhouettes do we have? Okay. Now, this is not easy to read. How about let's color the graphs? Okay. So what it does is, remember, 26, 9, 12, and 15. So the next step will help us color. So we want to plot X again, but now we want to plot the first 26 rows, 26 bars as dark green, not the next one as blue, the next 12 as yellow 2, and the next 15 as red. Okay. So this will give us better visuals. It looks nicer. So where do we get these colors from? These color codes actually come from a place called TK Colors. Okay, so you can go and look online called TK Colors. Okay. TK Color page tells you what are all the colors that you can have. So this is the RBG coding. Okay. So it has a lot of colors. You can even go and let's look at some of the diagrams. So this is the TK color chart. Okay. So you have multiple shades of grays. You have, my eyes just can't differentiate some of the colors. Right? For example, the gold and gold rod looks kind of similar to me, and definitely I cannot differentiate between turquoise and cyan. Okay. I don't know, some of you may be able to differentiate, but these are the names that you can use. So what happens if I make a boo-boo and instead of putting 26, I put 20. Okay. It basically just only colors the number of rows. So it colors 20 rows of dark green, followed by 9 rows of blue, and so on. And exactly, this is how it looks like. Okay, so you see, the blue actually gets shifted up, and then the green repeats itself again. Okay, so this is where you have to kind of remember these numbers as you go along. Okay. So here 4 groups seems to be working well. So let us see if 5 groups are okay. Let us just run the whole thing for 5 groups again. So we do the silhouette plot for 5 groups, and we put it into x.5, and then we replot it. So we do the same. Here, just show the color, assuming that after I plot, I have the number of rows. Okay. Okay, so x is not found. Have I lost x5? Yes, I forgot about x5. x5 has to start here. So I have to run the groups all over again for 5. Okay. So let me rerun the whole thing. Okay. So essentially this part, mnf.5, is actually the same command as this mmf.4 because it's rank 4. The other one is just rank 5. Okay. So you just repeat the same thing all over again. Okay, so again, it takes some time for this step. Let's just see. Okay. Okay, so now, yep. So this plot 5 groups. So remember 4 groups. Let's go back to the 4 groups in case you can't remember. So 4 groups, we have only 2 that goes into the negative. While 5 groups, we have quite a few. 1, 2, 3, about 8 that goes to the negative. So this tells us that perhaps 5 groups is a bit too much. Okay, so 4 groups may seem to work out well. So we are sticking with 4 groups from now on. Okay. Now, so mainly, personally, I think that 4 groups is better than 5 groups. So I'm quite disagreeing with Cool et al, saying that there are 5 groups of data or 5 cancer subtypes. So let us replot all this into our PCA plot. Remember our PCA plot, our first few plots. Just go back. It's pretty troublesome, but it doesn't go back that well. Anyway, so let's go there and we try to plot on our principal components. Let's say the first principal component, we want to group have the group 1, group 2, group 3, and group 4 into different colors. Okay. The main thing is in valid plot index, okay, doesn't like it. Over there. Okay. So remember this? So we want to plot it up, put the grouping into here to see whether can we see the 4 groups, are they overlapping? Okay. So this is how we run it. No, I actually did not want to run it this way. I want to run this. Okay. Now I have to wait for it, but okay, let me just wait for it to complete. Move yourself over. Okay, so just redid it again. Okay, so now what we want to do is to plot the data up for the principal components analysis and see whether are there overlapping groups. Okay, so let's run. Okay, so now you can see that the 4 groups seems to not overlap very much, except for maybe group 3 and group 4. Maybe it's group 3 and group 4. Okay, but they seem to be well separated. How about let's look at the first 4 principal components again and see whether do they match up what we see. Okay. So these are the first 4 principal components because it plots from 1 to 4. So let's run. Okay. Now you see that for most cases, it seems to quite separate quite nicely, except maybe between 2 and 3 and between 3 and 4. So there are some overlaps now. Okay. But 4 groups seems to be doing well. Another cool thing that we can do is, how about let's play around on a 3D plot. Okay, 3D plot, we actually plot between principal components 1, 2, and 3 on a 3 dimension. Okay. Then we can move it around. Okay, so this is what we do. You do a 3D plot 3D. Okay. And this gives you the output. So let me open it up. Okay. So this is where you can start to rotate around in the dimension that you have a good separation of all the 4 groups. So maybe this is a nice separation for all the 4 groups. Okay, then you can take an image for a publication showing that the 4 groups can be distinctly separated. Okay, so this is basically all that we have today. Okay, so something is wrong with my screen just now. Let me go back. So you just have a few more moments. Okay, so basically we have come to a conclusion to identify the tumor subgroups. So we identified that there are 4 tumor subgroups. Subsequent, we want to look at the differentially expressed genes. Okay, so as for now, we can kind of log out. So as we log out, okay, for example, when we close this, please always try to save your data. Okay, save the workspace. Okay, so now the workspace is being saved, and you can find that it's actually being saved as a workspace. Okay, so now this is the workspace that's being saved, your R data. Okay, so rather than say that it's R data, let's call this end of day 1. Okay, so we'll need this for the subsequent days because we want to bring back all the data again. All right, that's all for today. Hopefully that you are able to learn something from here. One thing I forgot to tell you is this practical or this set of practical is almost like a recipe for you to analyze future data sets as you come along. Okay, so you can use the same method to analyze it. It's just basically putting in a new set of data and rerun the whole script. So this is a very good set of data to have for you to rerun things. Okay, that's all for today. Have fun.